The aim of this procedure is to apply a stabbing injury to the ventral nerve cord of the Drosophila larval CNS and induce a cellular response to this injury. This is accomplished by first staging the larvae up to 96 hours after egg laying. Then, the larval CNS is dissected out and the ventral nerve cord is stabbed using a fine tungsten needle. The final steps are to culture and then fix the stabbed ventral nerve cords for immunostaining, or to just culture them for time-lapse recordings. Ultimately, confocal microscopy of the samples can show the dynamics of wound progression and changes in glial cell proliferation, apoptosis, and morphology.
The main advantage of this technique over existing methods, like injury to the adult fly, is that while it is still a paradigm for a fully functional central nervous system, the larval CNS is technically more accessible.
Generally, individuals new to this method will struggle because the ventral nerve cord can degenerate if the dissection step has not been mastered.
Begin by ensuring sanitary conditions. Clean the bench and your hands with 70% ethanol. The instruments should be wrapped in 70% ethanol-soaked tissue and set aside. When ready to handle the larvae, unwrap the tissue and allow the instruments to air dry. For these protocols, it is vital that the forceps tips meet perfectly. If they do not, adjust them using Arkansas stones or equivalent. Load one staining block with distilled water and 3 blocks with 2 ml of M3 PS medium. Select a vial with 96-hour-old larva